Friday, May 25, 11:30 in this room. What is this room? Merrill 12? Or are we Merrill 1? I always get these two rooms mixed up. I can't keep it straight in my mind. Looks like we're Merrill 1. So not in the laboratory room. And it's a 3-hour final exam like they all are. I don't think it'll take you 3 hours. It's worth 140 points. So what is that? That's like 1.5 times as big as about 1.5 times as big as a regular midterm in terms of points. Of those 140 points, about 60 are review questions on topics that you've already had midterms on. And the review questions are straight out of the textbook. Yes. Okay. So let me go over that real quick. If you look at the chapters in the textbook, at the end of each chapter is a section that's called review questions, and some of them are called the understand review questions, and some of them are called the apply review questions, and some of them are called the synthesize review questions. I drew review questions from all of those categories, but mostly from the multiple choice ones, which are the understand and the apply ones. But I reserve the right also to have gotten some from the synthesize review questions. Now, which chapters? Well, if you look at your syllabus, it spells out the exact chapters in the textbook that we've covered so far. It's mostly chapters 1 through 28. Although I think we skipped some of these, like I think chapters 24 and 25 and 26 are not in there. But anyway, any of the chapters that are listed here in the lecture topics are the ones you should be looking at for review questions. Does that part sound okay? Clear as mud? Okay, and the roughly the remaining 80 points are on topics that you have not had midterms on yet. And so those are going to include, those do include the following lecture topics: genomes, which we're probably going to not finish off today, but probably finish off on Monday of next week. DNA technology, this chapter we've been doing mostly in the laboratory. I'm probably going to end up starting a chapter on evolution next week on Monday and Wednesday, but I don't think we're going to get all the way through it. And there'll also be some population genetics problems. Basically using that Hardy-Weinberg equation that we've gone over once or twice. And that 80 points will also include some questions on the most recent laboratories we've done, which are transformations. That was the lab where you put the plasmids into the bacteria. Plasmid DNA isolation, which is the lab where you broke open the bacteria and took the plasmids back out. The last two labs we did, electrophoresis and restriction digestions. What an annoying noise. Still, it's better than chalk on fingernails on a blackboard. And the next two labs we're going to do next week, which are both on analysis of the digestions. Any questions? All right, let's see. Integrated in all this, I've integrated within it about 5 questions that I'm not going to be grading. You may say, well, why is he putting on questions that he's not going to be grading? Well, there's this requirement they have as for our classes these days called student learning outcomes, which are just some basic concepts they want all students who take a course to know. Like, what's the most basic thing that a chemistry student's supposed to know about chemistry, and what's the most basic thing an English student's supposed to know about English. And we have these student learning outcomes for this class also. And so they mandate that we put in these student learning outcome questions in our exams. So anyway, there's about 5 of these student learning outcome questions. I'm not going to tell you ahead of time which ones they are. That might bias you in some way, but know that throughout the test there's a few questions that I will not be grading. All right, and one last thing about the final. There's a lot of multiple choice questions. Most of these review questions are multiple choice questions, and some of the questions on the new stuff are also multiple choice questions. I think a little more than half the points, half of the 140 points are multiple choice questions. And so you're going to need a Scantron form, because I don't want to grade all your multiple choice questions by hand. So please pick one of these up before the finals day. You can get them at the bookstore. It is the 100 question multiple choice form. It's the brown one. And unfortunately, there's a green one also, but I prefer to use the brown kind. Is that your question? Yeah, get the brown kind. I don't know exactly how much they are, less than $1, I'm sure. Whew. Good stuff? Okay, so if you have any other questions on the final exam, come on down and see me after class and I'll try to answer them for you. Okay, let's hack away at this chapter on genomes. And the quiz that you'll have at the end of today's lecture is on the same chapter. All right, well, where we last left off was talking about the nature of the human genome. Remember, the genome means all the DNA that an organism has, including its genes and its non-genetic DNA, non-gene DNA. And this Human Genome Project was conceived in the early 90s, remember. And at that time, they'd certainly cloned several human genes, but no one had looked at all the human genes. Not all the genes had been cloned, so they didn't have a real global idea of what the human genome was all about. And so it was a pretty big task because if you add up all the chromosomes, it's around 3 billion base pairs in length. And sequencing technology was not what it was today. And so just to take on 3 billion base pairs worth of sequencing was a huge task. And so an international consortium of biologists was formed, and they subdivided the human genome among different institutes around the world. And it took around 2 years of just solid wall-to-wall sequencing, 24 hours a day. And I think I mentioned that the technology of sequencing DNA advanced as they were carrying out the project. And so there was actually a private company that tried it also. And even though this private company started later, they essentially finished in a dead heat with the international consortium. And so in the year 2000, they finished the sequencing. And so finally, they got a global look at what the human genome was all about. And there was a couple of surprises. one of them was that we had only 25,000 genes. They expected a larger number because they knew we had more proteins than that. And the answer turned out to be that most genes can undergo alternative splicing, or I guess I should say the primary transcripts of most genes can undergo alternative splicing. So yeah, we only have around 25,000 genes, but we might have 50,000 or even 100,000 proteins made from those genes. And here's the genome sizes of, just in terms of number of genes for some other species for comparison. So, you know, we're fairly complex organisms, certainly more genes than fruit flies or yeast or these simple worms called nematodes, but fewer genes than some plant species. All right, and when they got to looking at the actual sequences, and I'm sure they used computers to scan through all these G's and A's and T's and C sequences to look for start codons and stop codons and promoter sequences and whatnot. The major surprise that they found was how relatively small amount of the genome is actually genes, in meaning actual parts of the DNA that actually encode proteins. Only about 25%. Come on, laser pen, there we go. Only about 25% of the entire genome is genes. And when we say genes, we mean all of the gene, including the introns and the regulatory sequences like the promoters and the control elements and whatnot. And if you subtract that part out, because that's not really encoding proteins, all those are non-protein encoding regions, the actual amount that's actually codons encoding methionine amino acid and glutamine amino acid of the proteins is very tiny, only around 1.5%. So the amount of non-protein encoding DNA was a big shocker. So I want to discuss, well, what is this other stuff that our genomes are made out of? Why do we have so much DNA that's not all about encoding proteins? Well, they've subdivided the non-protein encoding stuff into various categories here. And this group right here is worth looking at, the non-functioning genes. What they find in around 2% of the genome are segments that are very, very similar in sequence to the actual functioning genes that we have, but are not expressing any proteins. Some of these are gene fragments where you might find the first part of a gene or the second half of a gene, but not enough to make the full protein. And in other cases, they have something that looks in sequence very similar to some of our functioning genes, but it has mutations. Maybe mutations in the protein coding region, or maybe mutations in the promoter or the control elements that make it non-functioning. Either in some cases the gene is never transcribed because the promoter might be mutated, or in some cases it might be transcribed but the transcript does not make a functional protein. And so these are actually thought to be sort of evolutionary relics. As a species evolves, certain genes essentially get cast aside because they're not being used anymore. That's probably what these are. About 5% of the human genome is what they call large-scale duplications. And a duplication means a segment of DNA that's essentially an exact copy of another segment within the genome. And about 5% of the genome are those duplications. And when they say a large segment duplication, they define that as 5 kilobases or larger. Remember, a kilobase means 1,000 nucleotides. So in other words, what they're defining this as 5,000 nucleotides in a row or larger. They find these multiple copies within the genome. And if you look at this diagram of the genome right here, this photograph of the genome right there, this red spot right here is supposed to be a segment of this particular chromosome. And let's see, in some cases, you find these large segment duplications at a different spot within the same chromosome. But in other locations, they're on different chromosomes. Like if this blue area right here represents a large segment here on this chromosome, you might find a duplicate of that on some completely different chromosome down here. And they're not 100% sure how these large segments get copied, get duplicated from one spot on the genome to another. And they're not really sure if it has any functional significance. You know, is this something that's good for our species or bad for our species to have big segments photocopied within the genome? They're not exactly sure, but there are some theories about it as to how we get these and what they do for us, and I'll talk about those here in a few minutes. All right, well, another type of DNA they find, and actually a fairly large chunk, are things they call tandem repeat DNA. And in your outline, you see it here. It's actually, this same type of DNA has a bunch of different names. It's sometimes called satellite DNA, not because they shoot it up into outer space or something like that, but when they were first analyzing the genomes, it would appear sort of side bands on like column chromatography type stuff. Anyhow,